now I get to spend the rest of this workshop telling you about a technology that we've developed at Mission Bio for very high throughput single-cell DNA sequencing that we believe will make the study of heterogeneity in tumor populations routine and accessible to clinicians and researchers. So the technology that we launched at the end of 2017 is shown here. It's our Tapestri Precision Genomics platform. It consists of 3 main modules. First, an instrument that runs disposable microfluidic cartridges. We also have consumable reagents that we sell as bundled 8-reaction kits. And those kits have all the reagents you need to go from either a cell suspension or a nuclear suspension to a sequence-ready library. And then because this high-throughput single-cell DNA sequencing data is quite complex, we've recognized that and recognized the need for easy-to-use and intuitive software solutions to process your data and properly analyze it. So we provide 2 software packages to complete our Tapestri solution. So the main product specs that enable broad applications across oncology are shown on this slide. Mainly, it's the ability for Tapestri to perform single-cell DNA sequencing in a targeted fashion on individual cells and very accurately call single nucleotide variants and indels across hundreds of targeted genomic loci. And we can actually get that genotype information across hundreds of loci on up to 10,000 cells per sample run. And that level of throughput ultimately affords us this ability to really go in and identify rare cell populations at or below 0.1% of the population. And the platform is now compatible with a variety of tissue types. Of course, for hematology, we have compatibility with PBMCs, bone marrow aspirates, but I'll tell you a little bit about some work we've done to make the platform readily accessible to solid tumor samples a little bit later in the talk. We have both cataloged panels that are available off the shelf for particular indications, as well as the ability to have investigators customize and target their sequencing to specific loci relevant to their particular application. I think Jerry already touched on this a little bit, but I just want to contrast our single-cell approach to sort of traditional bulk NGS, or next-generation sequencing approaches. With those approaches, you basically take an entire sample, so a population of cells, and you extract your genomic DNA, your nucleic acids, and you perform bulk next-gen sequencing on that population of nucleic acids. And what you get are variant allele frequencies across the entire population, but you really do not definitively know whether or not 2 mutations co-occur within the same cell or the same clone. And often there's quite a limitation in terms of being able to sensitively identify variants below a certain threshold. So this method often lacks sensitivity to call rare variants. So the Tapestri single-cell DNA sequencing approach is actually quite conceptually straightforward. We take individual cells and we confine them to droplets. And within those droplets, we have unique cell-identifying barcodes as well. Those barcodes get incorporated into amplified material from the targeted loci throughout the genome. And once that barcode incorporation has been performed, we can break the emulsion and do your traditional bulk next-gen sequencing on an Illumina platform. And using the cell barcodes, we can actually reconstruct the genotype of each and every cell within the sample to obtain this very definitive and accurate single-cell genotype information that can then be used to reconstruct the clonality of the entire tumor. This is a high-level overview of the workflow. Our workflow was purposefully made quite simple, and it's designed to seamlessly fit into existing NGS workflows that our customers are using. Again, you take a cell suspension or a nuclear isolate of interest, and you load that onto the Tapestri platform on the microfluidic cartridge. There, you can do your barcode reaction, and once that's complete, there's a very simple library prep step, and you're ready to go onto, again, Illumina-based platforms. and following your sequence information, data analysis, and visualization. So I want to take just a moment to sort of expand on what occurs on the Tapestri microfluidic cartridge, because that's really quite unique to what Mission Bio does, and it's really what gives us this really unprecedented capability to accurately genotype individual cells at such high throughput. So most droplet-based workflows people are familiar with are what I would refer to as single drop workflows, where you basically take your cells of interest and your reagents and barcodes, and you flow them all together and generate one population of droplets. That's great if you want to do RNA sequencing, but what we've discovered is that if you actually want to go after different genomic loci and very accurately assess them for variants, you actually need to use a different approach. And that approach we've developed is the use of a proprietary lysis reagent that contains a protease. So we flow this reagent together with individual cells or nuclei, and we encapsulate them into these picoliter volume droplets. There, the protease is able to not only effectively lyse the cell, but it actually digests nuclear proteins such as chromatin-associated proteins like histones that would otherwise act to sequester genomic DNA and hinder downstream molecular biology. Once we've used that protease to liberate the genomic DNA, we can then turn its activity off. And at that point, we have drops that have very efficiently prepared genomic lysate. Those drops can then be used on a second device where we then add in molecular barcodes, targeted gene-specific primers, and PCR reagent. The drops that are generated at that point are basically completely contained PCR reactors that can then undergo thermocycling and barcode incorporation into targeted loci can be done at that point. Once, of course, you get through the next-gen sequencing component of the workflow, you're ready to do data pipeline analysis and data visualization. And we have two modules basically to perform the different steps of the data analysis. The first one is our Tapestri pipeline, which first identifies cells from the next-gen sequencing data, and then is able to accurately call variants within those cells. Once you have the cells with the identified variants, you can upload that information into the second module, software module that we provide, Tapestri Insights, where you can very effectively look at co-occurring mutations and identify rare subclones and compare subclones from one sample to another sample. We've spent a great deal of time making our droplet-based reactions extremely efficient for robust detection of both copies of genomic DNA from individual cells. And that leads to really unprecedented performance across a number of key single-cell analysis metrics. We have a high degree of data completeness, a 10% capture rate. Again, the sensitive ability to detect populations at or below 0.1%. We have a very low allelic dropout rate, which is really essential to conclusively sort of identifying the subclonal architecture of tumor populations. We've worked in a number of areas to get our panel uniformity quite high, which ultimately reduced downstream sequencing costs. And we have low cell mixing and doublet rates from our platform. The platform has also been shown to be extremely reproducible in nature. Here we're showing 32 separate runs that were done in a very similar manner, where we looked at several different metrics, including throughput, panel uniformity, and allele dropout. And you can see that the variability across these sample runs is actually quite minimal. So the platform is really robust for doing this kind of analysis in a routine and scalable manner across many different samples. We've developed a robust pipeline for targeted sequencing panel development. This pipeline consists of first putting in different genomic coordinates or loci of interest and specifying those in our pipeline. We then have an algorithm that very efficiently designs primers to amplify up those regions in a highly multiplexed manner with a great deal of uniformity. And we, of course, once we've designed that panel, we manufacture and QC our targeted sequencing panels in-house. And that leads to this custom targeted sequencing capability that we offer, where you can design 20 to 300 plus plex amplicon panel, and our turnaround time is about 2 months for that process at this point. Additionally, we offer a number of off-the-shelf catalog panels. We have an AML panel, a myeloid panel, and a tumor hotspot panel, and we're continually adding to this list of catalog panels for different indications and applications. So with our current panels, we've gotten a lot of traction and applied our high-throughput single-cell DNA sequencing technology to a number of different areas: hematology, solid tumor profiling, and then gene editing or quality control for genome editing technologies. So I'll start talking about applications first on the area that we initially focused on, which was the area of hematology, and in particular looking at heterogeneity in acute myeloid leukemia. As Jerry mentioned, AML is known to be quite heterogeneous, and often the variants that are seen at the time of diagnosis end up being qualitatively and quantitatively quite different than what are observed from bulk sequencing at the time of relapse. This really points to underlying heterogeneity and evolution that's occurring within these tumor populations. And as Jerry mentioned, a large number of these patients relapse. 50% of patients relapse in AML. So understanding heterogeneity for this disease is likely to have benefit in the clinic. So we've worked with a number of investigators to show heterogeneity, aspects of heterogeneity for AML. I'm not going to talk about this particular work that we did with UCSF and Penn, mainly because I'm presenting it in another presentation at this conference on Friday. So if you're interested, please come to that. But I'll just very briefly summarize and say that we worked with our investigators, Kathy Smith and Sasha Pearl, to look at mechanisms of resistance to the FLT3 inhibitor gilteritinib. And what we were able to show with these collaborators was that oncogenic RAS mutations were occurring in the background of the FLT3 mutated clones, and that was really what drove this therapeutic resistance. And interestingly, we were actually able to look back earlier in the course of treatment before these resistance clones had really grown out and driven relapse. And what we were able to show in a number of patients was that we could actually detect extremely low levels of these oncogenic RAS harboring clones very early in the course of treatment, or even in clones that preceded the application of treatment. But I'll, again, since I'm presenting that on Friday, I'll just shift gears a little bit and talk about one other area in leukemia that we're applying our single-cell DNA sequencing technology and that we're very excited about. This is the analysis of minimal residual disease for AML. And we've done this work with a number of investigators, including Ravi Majeti at Stanford School of Medicine. He's actually on service this week, so he wasn't able to come present a more comprehensive picture of the study we're doing with him, but I'll just show sort of an exciting example patient from that study. So this patient actually was a 55-year-old female who acquired AML and then underwent standard of care chemotherapy and achieved complete remission at day 47. What we saw when we looked at diagnosis and relapse populations from this patient was the identification of 4 different variants in DNMT3A, IDH1, NPM1, and PTPN11. And the tumor burden was high, and a large part of the number of cells at these 2 time points contained all 4 mutations, so they were quadruple mutant. When we actually looked at day 47 remission, we were able to pick up 6 cells, and all 6 of those cells had all 4 mutations, so they were quadruple mutant, giving us high confidence that they're actually the leukemic cells that have not been cleared and actually ultimately led to this relapse. So this is, an area of research, the MRD, measurement of residual disease, that we're extremely interested in pursuing. But I'll shift gears just a little bit and talk about some work we're doing to enable solid tumor profiling. Much as we've shown, very interesting features with regard to genetic heterogeneity and leukemias, we feel like a lot of those insights are going to translate over to the analysis of the solid tumor world. And to enable solid tumor profiling on the platform, of course, we have our custom panels that people are ordering for specific cancer indications. But we also have a cataloged panel that contains 59 genes that are commonly mutated in different cancer indications. So we call that our tumor hotspot panel. One of the challenges with doing single-cell analysis on solid tumors is that you have to dissociate these different types of tissue and obtain individual cells to do single-cell analysis. And as we all know, different indications, different tissue types present different challenges and really require a lot of optimization to get single-cell suspensions. So what we've done is actually developed a really easy-to-use nuclear isolation protocol. We're not interested in the cytoplasmic RNA. We really just want to focus on the genetic heterogeneity in these populations. So we have an easy-to-use protocol that takes less than an hour that very efficiently isolates large numbers of nuclei from fresh frozen tissue. And we've shown that we can apply this nuclear isolation protocol across a number of different tissues: breast, lung, colorectal, prostate, melanoma, and obtain performance with our Tapestri platform that's virtually equivalent to what we see with our heme samples. And I'll just say that we also have in development compatibility with FFPE tissue. So we've teamed up with Charlie Swanton and his group at the Crick Institute in London, and we conducted an initial study, and I'll just show a representative patient from that study. Here we're looking at a patient with metastatic melanoma. And again, we used our universal nuclear isolation protocol on METs from different sites from this patient. And we were able to robustly get nuclei both from diseased tissue as well as normal tissue from these different sites. And this is just sort of a view of what it looks like when we isolate our nuclei and then stain them to quantitate. We aim for about 150,000 nuclei input into the platform. And once we have those nuclei and we load our cartridge, we run a workflow that's, of course, quite similar to what I've already discussed. The only difference being with our larger panels here to get the appropriate depth of coverage at our different targeted loci for the larger tumor hotspot panel, we use a HiSeq 2500 for greater sequencing depth. So this is just a summary of the run performance across the 5 different sites that were sampled from this patient. Again, we have a normal liver, a lung met, a liver met, and then 2 chest wall mets. You can see that the average nuclei or cell throughput from these runs is over 5,000 cells per run for 26,000 cells in total. Our panel uniformity is high, and we have enough coverage, sequencing coverage of all these amplicons to very effectively call variants from these samples. From this patient, across all the different METs that we examined, we found 5 somatic variants, 4 actionable, 1 that was a non-coding polymorphism or non-coding variant, and we found 26 germline variants. When we actually looked at bulk sequencing data that was obtained independently, we were able to show that the Tapestri sort of averaged VAFs match up extremely nicely with what was obtained for bulk sequencing, showing that we can call variants on par with what was obtained with bulk sequencing. So the correlation of those VAFs is shown here on the right. Of course, unlike with bulk sequencing data, what we can do is take our barcode information and then overlay that to identify the genotypes of all the cells within the sample. And once we've performed that analysis, we were able to identify 6 independent clones present throughout these METs. Once we had that information, we were able to sort of reconstruct a phylogenetic tree of what was occurring during this metastatic process. And what we saw were that there were two major branches that likely occurred off of an initial founder population that we can infer, since all the tumor cells that we identified had this BRAF mutation, as well as this non-coding SNP. Interestingly, one of the branches actually acquired a gain-of-function mutation in RAS, so an oncogenic RAS mutation at codon 13. And then subsequent to that, it appears that there was a secondary mutation that occurred in BRAF. On the right branch, this different evolutionary trajectory is shown, where instead of codon 13, codon 61 in NRAS is actually mutated. And then there's a transition to a number of cells either getting copy number gain at this position, or it basically just becomes homozygous. There's a secondary mutation. So there's two main trajectories that we've identified in the evolution of this tumor. And then we can take that information and sort of overlay that to the different regions to get the spatial information correlated with the genotypes for this patient. And what you immediately see is that the tumor cells that colonized the lung, so the lung mets that we sequenced, were largely or entirely composed of this left branch, having the G13 mutation and the secondary BRAF mutation. Conversely, when we look at both the chest wall mets and the liver met, we see that they were colonized by sort of that right branch of the phylogenetic tree, where they had the Q61 mutation, and then its conversion to homozygous status. It looks like, not surprisingly for the liver, we have, pretty much the entire portion of that sample was actually wild type cells or reference genotype. That actually ends up not being the case. There's a small percentage that was actually not wild type. And when we look at that a little more carefully, this is what we see. We actually, again, this is just looking at BRAF. When we look at the majority of those cells from normal liver tissue, of course, they're wild type for that BRAF sequence. But a small number, 8 cells, actually have the BRAF V600 to K mutation that was founding in terms of this melanoma patient. And so 8 cells in total had this genotype, and that's 0.13% of the population. And this is likely just an indicator of early colonization events or just cancer cells that ended up getting contaminated into this portion of the liver. But it speaks to the sensitive nature of our platform to really go in and find rare populations and accurately call variants and give you confidence that you're actually looking at either, you're looking at true tumor-relevant cells in that tissue type. So I'll just summarize the data we obtained with the Swanton group. We first saw that we had consistent and accurate sequencing performance on tissues of different origins with our nuclear isolation protocol. There was high correlation between our ability to call variants at the single cell level and the variants that were found from bulk sequencing data. As I just mentioned, we had a really nice demonstration of our ability to sensitively detect rare metastatic cells from normal tissue. And this data and some other patient data we obtained from Charlie's group is a motivated and expanded study that's currently in progress. So we're very excited about moving forward with that. I'll take just the last remaining bit of time in this workshop to talk about a final application that really sort of uniquely relies on our ability to do custom targeted sequencing panels. And this is our work that we've done with a number of partners to look at genome editing and its efficiency within individual cells. So I'll present this particular partnership that we did with a pharma partner, sort of an example of the types of information that single-cell DNA sequencing analysis with Tapestri can provide you that's just, again, completely missed with traditional bulk NGS. So this particular pharma partner was interested in looking at genome editing efficiency across 3 different loci. And not only did they want to know if these loci were edited, but they were looking for completion of editing. So they didn't want heterozygous edits. They wanted all 3 positions to have homozygous edits. And they also were interested in looking at off-target edits and potential rearrangements. So we designed a panel to interrogate these 3 different editing locations, and we performed Tapestri single-cell analysis to resolve basically all 27 edit combinations that are possible from the different efficiencies at these 3 different loci. And just to contrast that to the bulk sequencing data they had before they ran our platform, they had these VAFs at the 3 different positions. They have no idea if these are heterozygous or homozygous edits. They have no idea if they co-occur and if they've all occurred in the same cell. And clearly the editing efficiency is not super robust here. I should say we've run additional samples with them where they actually had really efficient editing systems and were able to get almost all the edits in a homozygous fashion and co-occurring in the same subclone. So with that, I encourage you to stop by our booth. Well, first of all, ask questions at the end of this, but also stop by our booth if you have additional questions. And please check out the oral presentation on Friday as well if you have time. So thank you.